Good evening. So yes, today we are going to begin with the chapter Cell the Unit of Life. So far we have completed what ma? Plant physiology. 3 chapters we have completed: photosynthesis in higher plants, respiration in plants, and then plant growth and development. Now we are going to begin with cell biology. Okay, along with plant physiology, which is the high weightage unit from your NCERT 11, is cell biology, in which we have 3 chapters: cell the unit of life, cell cycle and cell division, and biomolecules. From cell the unit of life, what are the concepts that we have to cover, okay, and from where we can expect the maximum questionnaire. First is cell discoveries and cell theory. From this mostly we can expect statement based questions, assertion, reason from the statements of NCERT, or then we can expect match the following type of questions. And then on shapes and sizes of cells, diagram based questions we can expect, and then we will be having prokaryotic cell, eukaryotic cell. In eukaryotic cell we have to learn the comparison between plant cell and animal cell, fine, and then we have cell membrane, okay, cell wall and plant cell wall and cell membrane. These concepts we will be covering today. Along with this, what are the concepts we have is, after cell membrane, we will be looking up to endomembrane system. Endomembrane system. Endomembrane system includes all the single membrane bound organelles whose functions are coordinated. Okay, like we have endoplasmic reticulum. In that again, smooth endoplasmic reticulum, rough endoplasmic reticulum. After that we have Golgi bodies, and then lysosomes, and then we have vacuoles. All the functions of this single membrane bound organelles are coordinated. Okay. So Zaira, we will have a plant kingdom class after cell biology. After cell biology, living world, biological classification, and then plant kingdom. Back to the concept, endomembrane system, and then we have double membrane bound organelles. Double membrane bound organelles include what? Mitochondria, chloroplast, and a nucleus. After learning nucleus, we will clearly look into structure of chromosome and shapes of the chromosome. Structure and shapes of the chromosome. Clear, right? Structure and shapes. So along with the single membrane bound organelles, with these organelles, we have to also look into microbodies beta. Microbodies, which are small single membrane bound organelles, which includes like peroxisomes and glyoxysomes. And then non-membrane bound organelles. We have to clearly learn and understand about ribosomes and centrioles. Along with all these concepts, most important concept, that is cytoskeleton. Cytoskeleton will include what? Proteinaceous filaments like microtubules, microfilaments, and intermediary filaments. These 3 things we will be learning, okay. And after this cytoskeleton, we will be also looking into cilia and flagella centrioles. Non-membrane bound organelles, ribosomes and centrioles. These are 2 non-membrane bound organelles we will be seeing. These are non-membrane bound. Okay. After this, cytoskeleton, which includes microtubules, microfilaments, and intermediary filaments. Amongst all these concepts, which are major hubs wherein you will get very frequent NEET previous year questions, are very good, prokaryotic cell, sometimes from the cell theory and from cell wall and cell membrane, most importantly from endomembrane system. Every alternate year definitely there will be a question on endomembrane system. Most importantly on shapes of the chromosomes, and with respect to mitochondria and chloroplast, the questions will be adjoined to your plant physiology from photosynthesis and respiration. Okay. Quite very few times you got questions on cytoskeleton, most of the times on the diagrams of, okay, microtubular arrangement in centrioles and cilia and flagella, you may get the questions, okay. And ribosomes too, you will get n number of times. Connecting with molecular biology also, you will get so many questions on the ribosomes. This is the complete gist of the chapter. Let us get started with the basic note that cell is called as basic structural and functional unit of life. Why? Because cell is the structure. First you have to understand the hierarchy of living organization, then you will understand why cell is the basic structural and functional unit of life. So if you see the hierarchy of organization beta, first atoms, which are the minute particles, first existed. Group of atoms which interact and they will form what? Molecule. Group of molecules will form what? Macromolecules, which forms out to be cell organelles. Till here, cell organelles to sustain, they are dependent on the other cell organelles. Like just now we have learnt endomembrane system where all the functions are coordinated. As such, be it non-membrane bound organelles, single membrane bound organelles or double membrane bound organelles, to survive and sustain, they are dependent on the functions of the other cell organelles. So, till cell organelle organization, there is no independent living, independent functional existence is not there. But a single cell includes what ma? A cell membrane and a nucleus with cytoplasm. Nucleus with the cytoplasm is called as what? Protoplasm. Nucleus with the cytoplasm is called as protoplasm, and protoplasm along with this membrane is called as what? Protoplast. Fine. This will form out to be one cell, and this is both structurally and functionally independent. Okay. Next, group of cells will interact. In living diversity we have learnt. Group of cells will interact and they will design the property of a tissue. Group of cells will interact and they will give the property of an organ. Group of organs interact, forming out an organ system, and organ systems will form out to be an organism. And if you consider the ecology, group of similar organisms will give you what? Population. Okay, group of similar organisms will form population. Different populations will interact to form what? Community. Different communities, yes, will form out to be an ecosystem. Ecosystems will interact, forming out what? Biosphere. Different biospheres will form out biome. This is, till here it is the hierarchy of living organization, and this will be the hierarchy of entire biome from single atom to biome. This should be clear in your mind. Okay. So in this, what is the first unit, basic functional unit, where cell is the basic structural and functional unit of the entire life, which has shown both structural and functional independence. It is both structurally and functionally independent. Understood, right? So now we will be looking into the basic cell theory. Before that, discoveries, right? Cell discoveries. Cell discoveries. So cell discoveries, if you have to look into, first basic cell discovery is done by what? Robert Hooke. First discovered by whom? Robert Hooke. Yeah. Here. Robert Hooke has discovered. Yes, Robert Hooke. He has identified or discovered the dead cells from the cork cells of Quercus tuber. Okay. He has discovered the dead cells from cork cells of which plant? Quercus tuber. I will let you know tomorrow. Okay, Quercus tuber is the scientific name. Fine. So here if you see Robert Hooke, he has discovered what? Dead cells, cork cells from the Quercus tuber. How did he identify these dead cells? Okay, as honeycomb structures without any protoplasm. Okay, as honeycomb structures without any protoplasm he has identified. Okay. Next is the discovery of a living cell. Living cell is discovered by Anton van Leeuwenhoek. Take down parallelly, ma. Yes, Anton van Leeuwenhoek, who is the father of microscopy, where he has observed different lens structures. He is a Dutch scientist. He has observed different lens structures, and that is how he has identified a living cell. like spermatozoa, okay, and symbol leaving cells like protozoans. All the unicellular organisms, most of them were identified by Anton van Leeuwenhoek. And lastly, it is Robert Brown. Yes, Robert Brown, who has discovered nucleus. Good. From the orchid root cells. From orchid root cells. Clear, right? So till here it is clear, Noma? Yeah. So let us explore one flow chart. One is in 1665, Robert Hooke is the first scientist who has observed dead cork cells, and he has given the term cell. From which cells he has observed? Cork cells of Quercus suber. And then in 1674, first Anton van Leeuwenhoek is the first scientist to observe what? Animal cells, mostly protozoan cells, protozoans and spermatozoa. And then Robert Brown in the year 1831 has discovered nucleus from orchid root cells. And then in 1838, Matthias Schleiden and Schwann has given us the cell theory. With the cell theory, I will let you know. First we will discuss cell theory in detail. So cell theory is given by two scientists. One is German botanist. And the second one is what? British zoologist. Fine. So who is that German botanist? Matthias Schleiden. Yes. And Theodor Schwann. Okay. In Matthias Schleiden, in the year 1838, he has said only one point, that all plant bodies are composed of cells and products of cells. He has given information only about plant cells, only one point he has said what? All plant bodies are composed of cells. Okay. Next it is the scientist Theodor Schwann, who is a British zoologist. In the year 1839, he has set out 2 points. One is all the plant bodies and animal bodies are composed of cells. This is the first point. Second is the limiting boundary of a plant cell is, cell wall. Limiting boundary of a plant cell is cell wall. Over the cell membrane, the limiting boundary of a plant cell is cell wall. Whereas in animal cells, the limiting boundary is what? Cell membrane. These are the points which are set by whom? Animal cells, limiting boundary is cell membrane. Fine. So these are the 2 points which are set by Theodor Schwann. Clear right now? So both of the scientists have failed to explain one point, that how new cells are existent. Okay, how new cells are formed. Both of the scientists have failed to explain. So how new cells are formed is being explained by whom? Rudolf Virchow. Under the concept of omnis cellula e cellulae. Omnis cellula e cellulae. This is called as modified cell theory. So whichever question will come to you on cell theory, you have to keep these 3 points in your mind. Raghav, sit straight. These 3 points in your mind. First is what? Matthias Schleiden said that all plant bodies are composed of cells. Second is Theodor Schwann, plant cells and animal cells. Both are composed, plant and animal bodies are composed of cells. Limiting boundary of a plant cell is cell wall. Limiting boundary of an animal cell is cell membrane. Fine. These are restricted to the cell theory. Modified cell theory is with respect to Rudolf Virchow in the year 1855. He has given us the concept of omnis cellula e cellulae, where new cells arise from the pre-existing cells. So here you see, ma? Yeah. First is Matthias Schleiden. So here observed that all plants are composed of different kinds of cells which form tissues. In the which year? 1838. In 1839, Theodore Schwann noticed that cells have a thin outer layer called as plasma membrane and concluded that plants have a cell wall, hypothesized bodies of animals and plants are composed of cells. Clear, right? And then it is Rudolf Virchow in the year 1855. He is the first to explain what cells arise from pre-existing cells, omnis cellula, e cellulae. Clear right now? Now, so cell theory is clear to us. So what are the three principles of cell theory? First is all living organisms are composed of cells and products of cells. Point number 2, all cells are from pre-existing cells. Third, activities of an organism are outcome of sum total of activities and interactions of its constituent cells. This point we have learnt. We have also learnt under cellular organization in our chapter diversity of living world. Okay. Now cell theory is clear to you. Modified cell theory is clear to you, right? Now we have to know what are the exceptions of cell theory. What are the cells or what are the organisms which are the exceptions of cell theory. First is virus, because viruses are just called as nucleoprotein structures. Okay. So here exceptions of cell theory. Exceptions of cell theory. First is what ma? Virus. Virus are said to be what? Nucleoprotein structures. Nucleoprotein structures or particles, nucleoprotein particles, or these are also called as infectious agents, or simply they are called as living entities. Why they are not called as a cell? Because they are just having an outer protein coat. They are just having an outer protein coat like this with the genetic material inside. There is no cytoplasm, there is no fluid filled space as such. Okay. So these are what exceptions to cell theory. Cell theory is applicable only to those cells where we have discussed now what are the characteristics of a cell. Those which has cell membrane, cytoplasm and a nucleus inside that, single nucleus. Okay, one nucleus should be there, cytoplasm should be there, which is protoplasm, and it should be covered by a cell membrane. Only to these cells cell theory is applicable. Now you can easily tell me the exceptions. So virus, along with virus, to viroids, to prions, cell theory is not applicable. And third is to those cells which possess anucleate condition. Anucleate condition. Anucleate condition is observed in few cells like what? Mammalian RBC cells, anucleate condition, mammalian, good, mammalian RBC cell, good, tracheids, vessels with, tracheids, vessels, most importantly sieve tubes. Sieve tube cells. To these cells which have anucleated condition or enucleated condition, enucleated condition, cell theory is not applicable. And then we have multinucleated condition, multinucleate or cenocytic condition, multinucleate or cenocytic condition. So multinucleate or cenocytic condition is possessed by many organisms. Like here you can see paramecium, which is a protozoan, will possess binucleate condition. Paramecium, binucleate condition, okay, wherein in paramecium if you take, okay, larger one macronucleus will be there and one micronucleus will be there. Macronucleus will conduct the activity of metabolism. It will control all the metabolic activities of the cell, and micronucleus, good, micronucleus will take over the reproductive activity of the cell. Fine. and few organisms like Opalina will possess multiple nuclei inside the cell, and the fungus like Rhizopus, which is bread mold, okay, this also possess xenocytic or multinucleated condition. Okay, so these are the exceptions of cell theory. These cells are the exceptions of cell theory. And then if you see different shapes of the cells, this is given in your NCERT, different shapes of the cells. We have what? RBC, which is round and biconcave, and do remember the progenitor cells will possess nucleus beta. Progenitor cells which are giving out RBCs, they will possess nucleus, but mature RBC will not have nucleus, which are round and biconcave. White blood cells are what? Amoeboid. Columnar epithelial cells are long and narrow. Nerve cell is branched and long. Nerve cell is the longest cell, remember. Nerve cell is the longest cell, and tracheid is elongated. Mesophyll cells are round and oval. Fine. So here you have to highlight three things beta. Longest cell is nerve cell. Largest cell is what? Ostrich egg cell. Ostrich egg cell. Single ostrich egg cell is the largest cell, and smallest cell. Smallest cell is mycoplasma. Mycoplasma. Mycoplasma are said to be the jokers of plant kingdom because they will change their shape according to the habitat in which they are going to infect. Okay, they are both plant and animal pathogens. They are facultative anaerobes. Fine. They are called as jokers of plant kingdom because they are pleomorphic. They will change their morphology or structure according to the habitat they are living in. Fine. Next, best example of mycoplasma is what PPLO, pleuropneumonia like organisms. Okay, and then is we are getting started with what? Prokaryotic cell. So prokaryotic cell here, if you see just a second, yeah. Before starting the prokaryotic cell, once look into different shapes and sizes of prokaryotic cell. We have 4 different shapes of prokaryotic cell. What are they? First is cocci, spherical shaped cocci. Singular form is what coccus. Okay, rod shaped bacilli. Singular form is what? Bacillus. Okay. Singular is spirillum, spiral shaped spirillum, plural is spirilla, and then vibrium, comma shaped vibrium, singular is vibrio. Singular is vibrium, plural is vibrio. This is comma shaped. Amongst these 4 different shapes of bacteria, which is more pathogenic? If that is the question, bacilli is more pathogenic. Take down parallelly. Yeah. So here we have different sizes of the cells. Eukaryotic cell is how much 10 to 20 micrometers in diameter. Typical bacterial cell is 1 to 2 micrometer in diameter. PPLO, which is the example of what? Pleuropneumonia like organisms, example of mycoplasma. Smallest living cell 0.1 micrometer in diameter. Viruses are around 0.02 to 0.2 micrometers in diameter. Clear, right? These are different sizes and shapes of the cell. Next we will get started with prokaryotic cell. So from prokaryotic cell what we have to learn? Okay, you have to start learning from the cell envelope, which is the outermost covering, then going to the cytoplasm. First you have to cover up the genetic material part, and then you look into the non-membrane bound organelles. What are the key characteristic features of prokaryotic cell? Nucleus is absent, that is wrong statement. Nucleus is present, but then you do not have a membrane bound nucleus. There is no proper membrane which is encovering the nucleus. There is no proper membrane bound organelle. Okay, that is the reason this genetic material is scattered inside the cytoplasm. This is called as nucleoid, not nucleus. Okay, that is one characteristic feature. Second is what? There are no membrane bound organelles inside the prokaryotic cell. Okay, no membrane bound organelles, and there is no membrane bound nucleus. Let us get started. So as how you see, like the outermost layer inside the animal cell, in an animal cell is what? Cell membrane. limiting boundary of a plant cell is cell envelope, but prokaryotic cell has 3 layered cell envelope as their outer boundary. Okay, it is what? 3 layered cell envelope. That 3 layered cell envelope, outermost layer is what? Glycocalyx. Yeah, outermost layer is what? Glycocalyx. Yeah, second is cell wall, third is cell membrane. Okay, glycocalyx can exist in 2 forms beta. Glycocalyx can exist in 2 forms. One is loosely arranged glycocalyx like this. Loosely arranged glycocalyx can be slime layer, and tightly arranged glycocalyx like this. Tightly arranged, that is capsule. Fine. So, the outermost layer is what ma? Glycocalyx. If it is loosely arranged, we are calling it as slime layer. Tightly arranged, we are calling it as what? Capsule. So, this slime layer, agar outer layer slime layer hai to, what is the benefit to the bacteria? It will help the bacteria in prevention of loss and minerals from its cells. Okay, it will help the bacteria from prevention of loss and minerals, loss of minerals and nutrients. Yes, whenever bacteria is exposed to either thermoacidophilic condition or halophilic condition in extreme environments, this slime layer will protect the bacteria from loss of water and minerals. If the outermost layer is capsule, okay, bacteria whenever it is in the host immune system, okay, this will be protecting the bacteria. Capsule layer will protect the bacteria from host immune system to survive in host immune system. Good, right? So, next, glycocalyx is clear to you. Most of the cell wall composition in bacteria will be peptidoglycan layer. Peptidoglycan. Peptidoglycan means what composition in the cell wall ma? Protein and carbohydrates. Okay, that is peptidoglycan layer. And then is what? Cell membrane. Cell membrane has some invaginations into the cytoplasm. Okay, these invaginations can be in the form of vesicles or in the form of tubules or in the form of lamellae. Okay, so the extensions, or you will call it as inclusions or extensions of cell membrane towards the cytoplasm. Okay, either in the form of vesicles, tubules or lamellae. Those extensions are called as what? Mesosomes. Here can you see the mesosomes now? Mesosomes are clear to you, right? So, these are the tubular extensions. One image that is drawn out is tubular extensions. These are mesosomal tubes, and at the center can you see the mesosome sac. What are the major functions of these mesosomes? Mark my words, many at times in your final NEET examination, questions asked on the mesosomes. Okay, the major functions of the mesosomes include functions of mesosomes. Take down now. Functions of mesosomes. Major functions of mesosomes include, first is cell wall formation, DNA replication, daughter cell formation, most importantly respiration, fifth function secretion, yes, last is increase in surface area. These are the functions of mesosomes. Amongst all these functions, the most prominent function is respiration. Okay, how respiratory function is most prominent is, they will release out the respiratory enzymes into the cytoplasm, which will help in oxidation of macromolecules inside the prokaryotic cell, because of which the mesosomes of a prokaryotic cell are said to be equivalent to mitochondria of eukaryotic cell. Okay? Mesosomes of prokaryotic cell, most important statement beta. Mesosomes of prokaryotic cell are equivalent to mitochondria of eukaryotic cell. Fine? To mitochondria of eukaryotic cell. Done with the functions of mesosomes? Any doubt anywhere beta? No, right? And then we will go to the cytoplasm. Now it is clear to you, right, with respect to cell envelope, outermost layer is what, Nana? Glycocalyx. It can be in the form of slime layer or it can be in the form of capsule. If it is slime layer, it will help the bacteria in prevention from loss of minerals and nutrients. If it is capsule, it is doing what? It will help the bacteria to hide from host immune system. Cell wall composition, mostly it will be peptidoglycan. Cell membrane will have some extensions. Extensions either in the form of vesicles, tubules or lamellae. Okay. So what are the functions of mesosomes? Cell wall formation, DNA replication, daughter cell formation, respiration, secretion and increase in surface area. Amongst all this, most prominent function is respiration. That is why here is a prominent note wherein mesosomes of a prokaryotic cell are equivalent to mitochondria of eukaryotic cell. Now here we have the genetic material. First we will look into the genetic material. Look into it, ma. So two genetic materials are present inside the prokaryotic cell. One we will call it as genetic material. We have two types. One we will call it as genomic DNA. Yeah, genomic DNA, which is also called as chromosomal DNA. Chromosomal DNA. Or the second type is non-genomic DNA. Non-genomic DNA, or which is also called as extrachromosomal DNA. Extrachromosomal DNA. Clear, right? So one is genomic DNA, chromosomal DNA, other is non-genomic DNA, which is also called as extrachromosomal DNA. So genomic DNA, what is its function, ma? It will look after all the metabolic activities of the cell. Okay, metabolic activities. Fine. Non-genomic DNA or extrachromosomal DNA is your plasmid DNA. Okay, this is the plasmid DNA. What are the major functions of the plasmid DNA? Plasmid DNA will give the specific phenotypic character to the bacteria, and the second function is to provide antibiotic resistance in the bacteria. These are the two major functions of plasmid DNA. And third most important function is it will help in sexual recombination by forming a conjugation tube between them. Okay, so what are the three functions, ma?
Phenotypic character.
Yes, first is what? It will provide specific phenotypic character. Phenotypic character to the bacteria. Why specific phenotypic character to the bacteria? It depends on the habitat in which it is living. Okay, whether it is in thermoacidophilic conditions or halophilic conditions or as methanogens or inside some other host. Based on that, specific phenotype character will be allotted by what? Plasmid DNA. Second is providing antibiotic resistance. Antibiotic resistance. And third is what, Nana? Yeah, sexual recombination, that is conjugation. Have conjugation by forming a conjugation tube. In that, in plasmid DNA, if at all the F factor is present, F factor, then it is called as F plus strain. So from plasmid DNA only F plus strain will be transmitted to the F minus strain. In this F minus strain also, inside the plasmid DNA only, the DNA will be transported. The F factor will be transported. Okay, F factor is being transported from which strain to which strain, ma? F plus strain to F minus strain through the conjugation tube, aiding what? Plasmid DNA. These are the three major functions. Plasmid DNA, many a times the questions are asked on it, do remember. Okay. Now let us look into the non-membrane bound organelles of the cell. Okay. Non-membrane bound organelles. So the first non-membrane bound organelle is your ribosome. First is ribosome. Ribosomes, which are discovered by George Pelliday. Okay, in prokaryotes the ribosome will be what? 70S ribosomes. What are they? 70S type of ribosomes with larger subunit 50S and smaller subunit 30S. Okay, what are the locations of ribosomes? You can see. Where do you find the ribosomes inside the prokaryotic cell? If you consider this as a prokaryotic cell beta, ribosomes are present attaching to the cell membrane and ribosomes are present inside the cytoplasm. Okay, those ribosomes which are present inside the cytoplasm are helpful in the protein synthesis. And those ribosomes which are attached to the cell membrane, they are helpful in what? Translocation of proteins. Fine, ma? So these are one non-membrane bound organelles. Second non-membrane bound organelles that you observe inside the prokaryotic cell is inclusion bodies. What are inclusion bodies? Inclusion bodies are storage bodies which are present in the prokaryotic cell. Inclusion bodies can be of phosphate granules, sulfate granules, synophysian granules. These are what? Storage bodies which are present inside the prokaryotic cell without any membrane. Okay. What are inclusion bodies, ma? Phosphate granules, synophysian granules, yeah, and sulfate granules. Yeah, good. Glycogen granules as well. Fine, right? Clear? And then, so with respect to non-membrane bound organelles, cell envelope and genetic material, everything is clear from prokaryotic cell, right? Now motile and non-motile structures of a prokaryotic cell, take them. Motile and non-motile structures, of a prokaryotic cell. First look into the motile structures, ma. The only motile structure that you see in a prokaryotic cell is flagella. Okay, flagella will be originating from the cell membrane and they are the extensions of the cell wall, and this will be your cell envelope. Okay, they will be originating from cell membrane and they are the extensions of cell wall like this. Okay, so what is adjoined over here? Basal body. It has three components. Flagella has three components. First is basal body, second will be hook, and then it is filament. Basal body, hook and filament. How the flagella will move? Not like a snake like this it will crawl. It will move like this. How? Because of the microtubular arrangement inside it. Of course, we will look into the microtubular arrangement in detail, but as of now you remember, in flagella it will have what, ma? 9+0 microtubular arrangement, wherein at the center there will be a proteinaceous hub. To this proteinaceous hub 9 radial spokes will be there. Okay, these 9 radial spokes peripherally they will be having 3 peripheral microtubules like this. Okay, so this will be rotating as such and the movement of flagella will be like this. Okay, so it will be showing what, ma? 9+0 microtubular arrangement. And what is the protein that is present on the flagella? What is the protein, ma? Flagellin protein. What is the what is the protein that is present? Flagellin. Remember, cilia is present in protozoans. Protozoans are what? Unicellular eukaryotes. Cilia is not present in prokaryotic cell. The only motile structure is what? Flagella. Understood, ma? Next is what? Non-motile structures. Non-motile structures include, first is pili and second is fimbriae. How can you describe pili and fimbriae? Pili can be described as the tubular extensions from the cell wall. What are the pili, ma? Tubular extensions from the cell wall. From cell wall. Okay. So, these will be the tubular extensions, and these tubular extensions will also act as what? Conjugation tube. Conjugation tube during the time of sexual recombination. As it is aiding in the process of sexual recombination, this is also called as excuse me, sex pili or true pili. Sex pili or true pili. And then we have what? Fimbriae. Fimbriae are nothing but the bristle-like structures which are sprouting out of the cell wall. Okay, these are bristle-like structures which are sprouting out of the cell wall. Their main function is attachment to the substratum. Attachment to substratum. So, both pili and fimbriae, they will aid in the attachment of bacteria to the substratum. Additionally, pili are also helpful in what? They will function as conjugation tube. So, they are called as true pili or sex pili. Clear, right? Now, we will be looking into two different types of bacteria. Those are gram-positive bacteria and gram-negative bacteria. Good evening to the online students also. Any doubts anywhere, you can message in the chat box with her. Enough. Yeah. Gram staining technique, which is developed by Christian Gram. Christian Gram. So, what is this gram staining technique actually? Okay. Look here, first we will understand the experiment. Two bacterial cultures were taken. Two bacterial smears were taken, and both of them were added with what dye? Crystal violet. Both of them were supplemented with crystal violet dye. Okay. After which both the bacterial smears will absorb the dye, right? Then they are washed out with alcohol. They are also treated with the mordant iodine. Fine. And then they are restained with, after decolorization, they have restained with safranin, but one bacterial culture has still retained the color of crystal violet, that is purple, whereas the other didn't retain this color, whereas this has shown what? Safranin color. Okay. To understand this better, you should know the cell wall composition of gram-positive and gram-negative bacteria. Then you will understand why gram-positive bacteria has retained the crystal violet color. Listen here carefully. First is gram-positive bacteria. It has 80% of peptidoglycan in its cell wall. 80% of peptidoglycan. Along with it, it also has teichoic acids and murin. Fine. A simple example I will give you with. You have taken a thick cotton cloth in which cellulosic fibers are very thick like this. Whereas a very silk cloth or polyester cloth whose fibers are not much thick. Both of them you have dipped in a dye. Okay. And then you have observed for the color. Which will stain more? Cotton cloth will stain more. Even after washing with normal water also, the stain doesn't go on the cotton cloths. Why? Because the fibers are very thick and it has absorbed the dye too much when compared to the silk or polyester. Same the case here, peptidoglycan content is more in the gram-positive bacteria, which is around 80%. Okay. It has teichoic acids and murin. Okay. Wherein this absorbs the crystal violet dye very tightly. Whereas gram-negative bacteria, this just has 20% of peptidoglycan and there is no teichoic acids inside this. No teichoic acids, no murin, we will call pseudomurin here. Then what does gram-negative bacteria contain? Polysaccharides and lipids. Polysaccharides and lipids. okay, which are loosely arranged like this in the outer membrane. So they don't absorb the dye so much. Okay, when they are destained, it gets destained and absorbs this dye of what? Safranin. So with this, what did we observe? These gram-negative bacteria can adapt in the any conditions because of its cell wall of less peptidoglycan, more polysaccharides and lipids, and there is no murin here, no teichoic acids. Fine. So most of the pathogenic bacteria will be gram-negative bacteria only. Fine. Understood? Most of the gram-negative bacteria are what? Pathogenic bacteria. Most of gram-negative bacteria are pathogenic because of its cell wall. Okay? Understood? Understood the experiment and understood why gram-negative bacteria has very, why is it not retaining the color of crystal violet. Clear, right? Staining process, staining technique, and about gram-positive and gram-negative bacteria. Yes? Best examples for gram-positive includes Clostridium, gram-positive includes Clostridium, Bacillus. Bacillus comes under gram-negative. Clostridium you take as of now. Gram-negative includes Rhizobium, and most of the infectious bacteria like Salmonella typhi, all this comes under what? Gram-negative. Okay, clear, right, ma? Next, about eukaryotic cell, we have two types of eukaryotic cell, plant cell and animal cell. So first look into the organelle in detail. Today we will be discussing about what are the organelles like non-membrane bound, single membrane bound and double membrane bound organelles present in both plant cell and animal cell. And from tomorrow we will be starting with the discussion of cell wall, cell membrane, endomembrane system, double membrane bound organelles. So here you look into first in this diagram. Yeah. First the outer envelope is what, ma? Cell wall in a plant cell. Okay, in a plant cell the outer envelope is cell wall. And then we will focus on the double membrane bound organelles first. What are the double membrane bound organelles? Nucleus. Nucleus will be having four components: nucleolus inside it, nucleoplasm, nuclear envelope, and here nuclear pores. Second double membrane bound organelle is what? Mitochondria. And third is what? Chloroplast. These are the double membrane bound organelles. And over the cell wall there are cytoplasmic connections called as what? Plasmodesmata, which will connect the cytoplasm of two different cells. And then we have what, ma? Single membrane bound organelles. Extensions of this nuclear envelope will show us both rough endoplasmic reticulum, over which ribosomes are there, and smooth endoplasmic reticulum. Fine. Second, single membrane bound organelles include what? Vacuoles, Golgi bodies, and lysosomes are least present over here. And 90% of the plant cell's volume is occupied by what? Vacuole. This vacuole is called as what, ma? Sap vacuole, which is covered by tonoplast. Sap vacuole covered by what? Tonoplast. Okay. And then we have what? Small microbodies, which are peroxisomes and glyoxysomes. Then we have microtubules, cytoskeleton structures. Microtubules, microfilaments. In the diagram only microtubules are given. And the only non-membrane bound organelle that you see in a plant cell is ribosome. This is the only non-membrane bound organelles. Rest is what? Beta? Cytoplasm. Clear with all the organelles labeling? Clear, right? Now we will exercise all the non-membrane bound organelles, double membrane bound and single membrane bound. You have to tell me. First, non-membrane. The only non-membrane bound organelles you have observed is what? Ribosomes. Remember, we have two types of ribosomes. Both 70S ribosomes are present, 80S type of ribosomes are present in plant cell. 70s, you know, larger subunit is what? 50s, smaller subunit is 30s. 80s type of ribosomes, larger subunit will be 60s, smaller subunit will be 40s type of ribosomes. Okay, what are the locations of this ribosomes inside the cell? Locations. Inside the cytoplasm, you will find 80s ribosomes, and the extensions of the nucleus, extensions of the nuclear envelope, we have ribosomes, right? These are also what? 80s, which are present on the rough endoplasmic reticulum, and those ribosomes which are present in chloroplast are 70s, and those which are present in mitochondria are 70s. These are the locations of ribosomes, beta. 70s you will find in chloroplast and mitochondria, 80s type of ribosomes you find in cytoplasm and over rough endoplasmic reticulum. Clear, right, ma? Second is what? Single membrane bound organelles. Single membrane bound organelles. See. Single membrane bound organelles. Single membrane bound organelles, we have what? First is those included under endomembrane system, which includes endoplasmic reticulum, Golgi bodies, lysosomes are least present, and then vacuoles. Fine. These are present in the endomembrane system. And we also have what, ma? Microbodies like peroxisomes and glyoxysomes. These are single membrane bound organelles. What are the double membrane bound organelles that we have come across? Double membrane bound organelles. Double membrane bound organelles include nucleus, yes, chloroplast. Third is mitochondria. Nucleus, chloroplast and mitochondria, these are double membrane bound. Clear with the organelles here? Next, animal cell. Animal cell, which is given in our NCERT, this is the diagram of, yes, Raghav is here. Who is this? Yeah, animal cell. Here we have what? This is the organelle of, this is the cell which is given from the small intestine. Listen, Raghav, focus. As microvilli is there, we can say this is a cell from small intestine. Okay, and the limiting boundary is what, Ra? Limiting boundary is cell membrane, because it is an animal cell. Here we have plasma membrane and microvilli over here. And double membrane bound organelles include only mitochondria and nucleus. Nucleus, again we have nucleolus at the center and nuclear envelope. Now looking into the single membrane bound organelles, we have smooth endoplasmic reticulum, Golgi bodies, rough endoplasmic reticulum, lysosomes, and vacuoles are negligible in the animal cell. Lysosomes negligible in the plant cell, and vacuoles are negligible in animal cell. And then we have peroxisomes. Fine. Non-membrane bound organelles, along with ribosomes, we also have what? Centrioles. Centrioles will aid in the process of what? Cell division. Through the formation of spindle apparatus. Clear, right? Now, let us rewind the organelles which are present in the animal cell. First, non-membrane bound, what do we have, ma? Yes. First, it is always we have to consider ribosomes. Yeah, two types we have, right? 70s and 80s. Larger subunit is what?
50s, 30s.
Here again?
60s, 40s.
And 40s. Fine. In what type of, where are the locations of 70s ribosomes? In mitochondria. We have 70s in mitochondria, and in the cytoplasm we have 80s, and over the extensions of endoplasmic reticulum. Here also we have 80s type of ribosome over RER. Clear, right? And then after this we have second non-membrane bound organelle is centrioles. Done. And then we have Single membrane bound organelles. Single membrane bound organelles. Yes, we have what? First, single membrane bound organelles, we have to remember the organelles of what? Endomembrane system. Many times in your NEET, I think quite a times, 2 to 3 times, just the question is placed on whether these organelles are present in endomembrane system or not. What are the organelles involved in endomembrane system? Such a simple questions are asked. So that important it is. Endomembrane system, RER and SER. And then we have Golgi bodies, lysosomes and negligible vacuoles. Okay. Then we have what? Microbodies like peroxisomes, microbodies, only peroxisomes here in the animal cell. Okay. Next is what? Double membrane bound organelles. We have only mitochondria and chloroplast. Mitochondria and chloroplast. Sorry, yeah, mitochondria and nucleus. Clear, right? So these are the double membrane bound organelles that are being observed in the eukaryotic cell. So with this we will end up today's session. In our next session we will complete cell wall, cell membrane and complete endomembrane system. Okay, cell wall, cell membrane, endomembrane system will be done in the next class. So that's all for today's session. Any doubt anywhere you can ask me anytime. Okay, and you online students, you can message in the chat box beta. Thank you.